Hello? Okay. So we are going to get started on session 4, zoo research and engagement. This is the last presentation before we go to lunch, and then we will be back for the continuation of session 4. So I am happy to introduce our previous research intern. She just finished up last year. She was studying a pair of red pandas, George and Kuzi, in our park. So Marjon Joustra researches animal behavior and welfare in captive wildlife populations. Her work examines how environmental factors, including visitor events and lighting conditions, influence behavioral patterns and welfare indicators in zoo animals. So Marjon, whenever you're ready. Okay.
Yeah. Now, welcome to this presentation about the behavioral patterns and effects of the Light Festival on the two red pandas in the Serengeti-Park. This research is aimed to study the social behavior of the activity pattern of the red pandas. This includes the behavioral pattern, the breeding behavior, how they utilize the space, the interactions between the two, and the influence of the weather in the Light Festival. And the data was collected between 13 October and the 28th of November. Red pandas are herbivores that live in temperate forests with bamboo understories at an altitude between 1500 and 4800 meters. And they live solitary lives and are nocturnally active. In the IUCN Red List, they are classified as endangered. For this study, I have set up 5 hypotheses. First, the activity pattern is influenced by the weather and the temperature. There's an increase in activity levels during the night. The social interactions increase as I get closer to the breeding season in January. The physical distance will stay consistent during the study period. And lastly, during the Light Festival, there will be an increase in locomotion and exploratory behavior. This study consists of 2 individuals, a male and a female. And for the day observations, I used a focal sampling and scan sampling method for a 7-week period. And night observations were done using cameras and a similar observation method. Thermal imaging was used to get an average of the body temperature. And below the scores are shown to that I used for the weather scores. Here are the statistical tests that I used. The multiple linear regression is used to compare the temperature and the weather conditions to the activity. The Wilcoxon rank sum was used to compare the activity pattern during the day and the night. The Kruskal-Wallis method was used to compare the amount of time spent on behavioral patterns and the distance between the two red pandas over the 7-week period. And lastly, the Spearman rank correlation is used to examine the amount of time spent in locomotion during the Light Festival. And here you can see the placements of the 2 cameras that are the pink dots in the corners and the area of which each camera can see. And this is the heat map of the distribution of the red pandas in the enclosure. Here you can see that they prefer the platforms on the left and the tree right next to it. The average surface temperature of George was 18.8 degrees Celsius and of Kusi was 18.03 degrees Celsius. And below in the graphs, you can see the percentage of each enclosure, the enclosure use per individual. And here you can see that George was seen more on the platforms compared to Pussy, and she was seen more in a tree and walking on the ground compared to George. And here's the distributional behavior, the behavioral distribution shown per individual. Most of their time they spent resting, and both almost spent the same amount of time sitting, while George spent more time in feeding behavior and Goosey more in walking. And this is the results from the first hypothesis, from the weather and the activity patterns. Kusi showed a significant increase with rising temperature for vigilance and resting, and a significant decrease with rising temperature for social resting, exploring, scent marking, and walking. And a significant increase with rising weather scores for scent marking, and a significant decrease for resting. And George showed only significant decrease for social resting and normal resting for rising temperature. Here are the results for the second hypothesis. This is where the significant difference in activity shown during the day observations compared to the night observations. And social behavior showed for both individuals no significant difference over the 7-week study period. During week 4, there was a drop in social behavior, which also coincides with the start of the Lights Festival. Over the time of the 7-week study period, the mean distance between the red pandas increased, but not significantly. And the mean nighttime locomotion during the Lights Festival was significant for both individuals in locomotion and one individual in locomotion. So the strong preference for elevated structures supports their natural arboreal behavior in the wild, while the predominant resting behavior also coincides with previous studies on the captive red pandas. The weather only had minor effects on the behavior of the red pandas, and there's a clear night and day difference when it comes to the activity patterns. There was no significant increase in social behavior during the study, which also coincides with the solitary pattern of the red pandas, the wild red pandas, and the distance between the two showed no significant increase. And during the Lights Festival, the nighttime locomotion increased, which would indicate a subtle behavioral impact. But overall, the behavioral pattern was stable and is typical for the species. The behavioral patterns shown during the study are largely consistent with the wild counterparts, while they're still reflecting the captive management and the environmental conditions. This study does contribute to a better understanding of the captive red panda behavior, although it is still a baseline study that allows the park to evaluate their social behavior during the mating season. For future research on the behavior of the captive red pandas, it would be helpful to evaluate the behavior into the mating season. Also, multi-zoo cooperation for sharing data and conducting research on red pandas for a bigger understanding of the species. Furthermore, doing continuous monitoring for movement, proximity, and nocturnal behavior, for example, using video cameras. And would also be helpful to include hormonal analysis for continuing the assessment during the Light Festival for a better understanding of the effects of the artificial lights. And I want to thank Dr. Idul, Dr. Corina, Philip, Ruth, and the zookeepers and the IT specialists for their feedback and the support during this project, and for making it possible for me to complete this research. And I want to thank you all for listening, and are there any questions?
Hi, thanks for the talk.
Hi.
I just wanted to ask, why do you think during the Light Festival they were more active, but they're nocturnal? Why do you think that they were more active when there was more light?
Most likely because I think that they were just disturbed a little bit because of the light at a part of the enclosure. I can go back a little bit to the maps. There. Because here at the southern side, there was light, and well, you can see that they weren't there in general a lot, but they can still be affected by the light, because normally it's really dark at night. So how do I say this? Yeah, they will be so most likely that they're just disturbed from the light in general. Yeah.
Okay, thank you.
Are there any other questions for Marjon? Okay, so if I may, with regards to the question, I think that the animals were generally more active because with the brightness of the lights, it was akin to being like daytime almost. And yeah, and that ties in as well to when they're cuddling in the evenings and just watching the lights and the people go by, because we have the cameras placed there, so we're also able to observe them at nighttime, and they're usually together in the evenings, and then they run around, get some food, and then come back again together. So I think it was the novelty of the lights and yeah, activities that were related to the day was similar to what was occurring in the night. Yeah, I guess the question would be more, would you consider it a positive welfare to have these event lights, or would you say it's more neutral or a negative impact?
Yeah, from what I've seen over the period, it's just in the beginning that there was a difference in the behavior, and after that it just started to be more stable again. So I think over time that they will get used to, again with it.
Okay, thanks, Marjon. Are there any other questions? All right, so we're gonna, if we can, please give a round of applause to Marjon. Thank you very much. Now we are going to have some lunch and then come back and continue with the session 4, continuation, zoo research and engagement. Oh, sorry? Oh, yes, and then we have the group picture after lunch. 1:40. At 1:40, okay. Where? Just outside. Thank you all. Thanks, Marjon.
Thank you.
Let me know when. Okay, welcome back, everybody. So we will continue with session 4, zoo research and engagement. Our next speaker is Dr. Ellen Williams. She is an expert in animal behavior and welfare, particularly in zoo settings. Her research includes welfare assessment and the role of zoos in conservation. She is actively involved in professional organizations, and she contributes to animal welfare advisory work. So please join me in welcoming Dr. Ellen Williams.
All right. Hi. Thanks, everybody, and thanks very much for inviting me. So I'm very excited to be here. I'm going to talk to you today a little bit about the possibilities of using citizen science to actually engage the public within zoos, and thinking about how we take them from just being a visitor to being a volunteer that can contribute data. So I'm probably preaching to the converted, really, but we need to make sure that we engage the public. This is a bit of a sad story, but this obviously is the case. So we are down 73% on our Living Planet Index, and this is only going to get worse as time goes on. And when we look at the threats to biodiversity, what we see is that the public are within all of these, okay? So we've got changes in land and sea use, which means that habitats are becoming fragmented, animals aren't able to get different places, we're over-exploiting species, be that for food sources or for other means. We've got pollution, we've got microplastics everywhere, we've got invasive species that we're mostly responsible for letting out into the open, and then we've got climate change exacerbating all of this. So if we look at this Humans then are the crux of the problem, so we need to engage humans to try to find a solution. And then if we think about this from like a One Health point of view, because if we want to engage people, if we want to get people on board, I think it's important that we understand where they fit within this model. So from a One Health point of view, human health, animal health, ecosystem health is intrinsically linked. So they play a really important part in this. Obviously, the theme of this conference, engaging minds, saving species. Traditional research is not enough to do this, okay? We've got a really serious problem, and we haven't got enough researchers on the planet to solve this problem. We can't collect all the data that we need to just as researchers alone. So if we can start to think about engaging people to help generate data, then we've got lots of opportunities. And zoos have got a really sort of well-placed to do this. So in a nutshell, what is citizen science? It's the process of engaging people, okay? It's a process of kind of sort of collaborations between researchers or scientists and non-scientists to generate data. And we've got these sort of two aspects. So we've got the citizen science side, which is when people collect useful data. And then we've got the engagement side. And both of them are equally important, and zoos can equally do both. So there's lots and lots of examples of citizen science when we look at like the wildlife conservation literature, lots of kind of tell us what you've seen, tell us where you've seen it. There's a little bit more if we look at like the freshwater watch. So this is getting people to actually collect data from aquatic areas, so from streams and things. They get a pot, they send the pot off, and then that tells us about ecosystem health. This wildlife trust stuff is about, a woodland trust, sorry, is about telling us are trees starting to bud sooner than they were previously. So we've got that kind of telling us a little bit more about climate change, the impacts of climate change. Yeah, okay, so and then here's just a couple of examples of data that's actually been collected by citizen scientists. So this was a project in Java and Bali, and it's looking at monitoring and distribution of bird species. So, I mean, bird people are mad, right? You all know. You bird people know that you are mad. And as soon as you've got an opportunity to create a list and send that in of things you've seen somewhere, you are right on it. And so what they had was, there were over 20,000 lists with over 100,000 bird records. So this is really incredible at giving like a baseline species distribution. And this is work that's been done on camera traps and image analysis from those camera traps. So this is a monitoring system across Europe. And the idea is that they can monitor biodiversity change and also over space. So how are animals using space, and also biodiversity change over time. And that starts to just tell a bit of a story about what's happening in the wild. Camera traps are amazing, right? If you can get funding for camera traps, perfect. But you've then got to find the time to analyse those camera traps, unless you can get a model to do it for you. So actually engaging the public in something like this, it gives them a job, gets them engaged, and then also you generate amazing data. And then this was another project that was looking at grey seals. So this was more of a tracking project. This was done on a program called EpiCollect 5. Basically, they wanted people to give them information about seals that they'd seen, what the seals were doing, where they were. And this also, so they wanted to look at the seals and what was happening with the seals, but they also then wanted to look at who is engaging with this process. Because if you know who's engaging, you can work out who you can then target or who you still need to target. So there's kind of like a double benefit there. And then if we think about this in a zoo context, so around the world we've got in the region of 8 billion people. Not all of those people are citizen scientists. They don't all want to be citizen scientists. One hopes that in a zoo you've at least got people that care a little bit about animals. So maybe we're kind of pushing at a slightly open door. So this is just some examples from British and Irish zoos. Lots and lots of different projects that have been undertaken. Many of these projects who are focusing, while brilliant, are focusing on kind of tell us about what you've seen, but you're not really focusing on tell us about what you've seen within our actual collection. It's more kind of like, have you seen sort of native species whilst you've been on site? What have you seen and where have you seen it? So what we can do instead is start to think about, can we engage the public with the animals that we are managing, the animals that we're keeping? And we know from research that if people are engaged with behavioural monitoring, it kind of gives them a little bit more of a, not a relationship, but kind of they think more about that species. We know it starts to build empathy, and we know that can relate to pro-conservation behaviours. So we wanted to see if we could actually apply this within a zoo setting. We've also, if we think about the role of the modern zoo, I mean, this is obviously ever-changing, but there's lots of different aspects to it, and if we think about where citizen science fits, it's contributing really important scientific research. It is importantly potentially contributing education and training, because if you're telling people how to do a behaviour observation, they're learning about that. But if you're increasing your dwell time at your exhibit, you've also got opportunities to train them in other things whilst they're there. And then importantly, you've got this public health and well-being aspect as well. And I'll mention that in a minute. Oh, the thing's come on, so I know what's happening next. That's exciting. Good. So the aim of this study then was just to provide visitors with an opportunity to engage in an animal behaviour research project at a zoo. As a kind of a sideline, we wanted to know if we could use this data, if this data was going to be beneficial, but the most important thing for us was, will visitors engage to begin with? So we had 2 study locations, both in the UK, one smaller zoo, Fernbell Conservation Project, and then one, as Pip and Sarah were saying earlier, middle-sized zoo, Twycross Zoo, and we were looking at the Asian small-clawed otters and Rothschild giraffe. And we chose these species because, A, we wanted species that we thought people would be interested in and that would be doing something. And then we wanted to think about if visitors are flocking to do our study, because we're like, they're going to flock, right? They're going to flock, so we need space for them to do that. And we wanted to have somewhere where we could put up video cameras. So at Fernbell, they had just a QR code. They had to scan the QR code. And then at Twycross Zoo, I encouraged, is the word we'll use, to encourage visitors to scan the QR code and then take part in those behaviour observations. And so they were supposed to watch for 2 minutes. Whether or not they did, who knows. But they're supposed to watch for 2 minutes, and they had a 4-part questionnaire. So they told us what the animal was doing. They then undertook a qualitative assessment of that animal's experience. We asked them questions about welfare. So this was a thing we were super interested in, was what does welfare mean to them? How do they define welfare? And then also, where have they got that information from? And then the last aspect was the citizen science process. So how did they find that process? Did they enjoy it? Would they engage again? I'm going to focus on these 2 today just for time, but I'm very happy to talk about all of it if anybody wants to catch up about this later today. Please do feel free to take any photos that you want, but I'd be grateful if you didn't share it, because we're still trying to publish this paper. It's been in review for what feels like forever. But yeah, we might sing happy birthday to it soon, but we'd be grateful if you didn't share it on social media. So the first thing we wanted to know was, will people actually engage with this process? And importantly, will they engage if they're not being harassed? So that's this zoo here. And what we found was actually, yes, they will. So on average, we had 2 to 7 observers per day at Fernbell and 14 to 41 at Twycross Zoo. So that was good. That was great they actually engaged in the first place. But what we then wanted to know is, if you had this opportunity again, would you engage again? And the majority would. So as you can see here, over 90% would, and nearly 80% and over 80% found the process rewarding. So this felt really important to us, because even if everything else didn't work, like even if the data they collected wasn't usable, and even if they didn't sort of know much about welfare, the fact that actually they wanted to engage was really positive, because it means that they are open to these different ideas. We then looked at, so we gave them sort of open text, which was, on reflection, that was a brave choice, but we gave them open text to ask them what they knew about welfare. And then we coded it roughly, I'm sure you guys are all familiar with the 5 domains model, but we coded it roughly to that model. So we had the kind of the physiological domain, so nutrition, health, living environment. We then had the behavioural domain, so that was where, if they were saying things about like what the animal was sort of doing, how it was interacting with things, and then that kind of overarching mental state. And so first off, they actually knew stuff about welfare. So we were genuinely super excited by this. So the fact that they'd engaged with this process and then they knew a lot about welfare was really good news, because it means that visitors are mindful of, at least they're thinking about what welfare is. And if we can start having conversations about welfare, we can start getting people to be more mindful of interactions that they're having sort of more broadly. So the majority of time they were focusing on those kind of sort of the more input domains, so things like natural surroundings, hiding spots from crowds, large enclosures. We were seeing mental state mentioned, which was good, so are the animals, say, comfortable and content? And behaviour was talked about, but not as much, so that's when they were sort of saying, you know, the animals should be able to express natural behaviours, for example. And then, this was fabulous. Where have you got your knowledge from? The majority of people said they got it from a zoo. Whether that be the zoo they were in then, or a zoo, another zoo, the majority of it was coming from there. It was also coming from other places as well, but the fact that actually they're open to information being given to them within a zoo gives us a real platform to start having some really important discussions. So what does this all mean? So visitors were open to novel methods of engagement. They were open to actually doing the kinds of citizen science projects that we sometimes see in conservation actually within a zoo environment. We also see that learning then is happening in zoos. And if learning is happening in zoos, that means that we can start to change the methods, change the narrative, of what we're delivering to a zoo visitor. If they are taking things in within a zoo, we've got lots of opportunity to kind of get across really important messages. It's really likely that citizen science has got benefits for conservation education, even in terms of just getting people to be more invested in this process. I should have said earlier, I forgot to say, when I was at Twycross Zoo, I had a lady come up to me and she thanked me for putting this project on. And I was like, well, that's weird. Like, why is she thanking me? A bit nervous. And she said, oh, I've got a neurodivergent child and she's never spent 2 minutes standing and looking at something. And you being here today has meant that she is engaged in this process. And I was like, well, that's like, gosh, I was like, well, that's quite emotional. Thank you, you know. But actually, really, really important, especially when we think about maybe some of those less represented user groups that we do still have in zoos. Despite them, so they knew a lot about welfare, but we were still seeing this kind of this focus more still on those physiological indicators, more those input measures. I mean, it's good that they're recognising those, but potentially we maybe need to branch our education out a little bit and start thinking more about, okay, are we actually getting visitors to think about behavioural interactions? Not just have we given the animals some food, but actually is the animal engaging with that food? Or not just has it got enrichment, but is it using that enrichment? But the fact that we're actually considering mental well-being is hugely positive. And I've just come from the IMZRCB conference, and we were talking about like the kind of affective state stuff. And, you know, 20, 25 years ago, when people started thinking about some of these things, you would just be laughed at if you were talking about affective states. So it's super important as an industry, we're now recognising how important mental well-being is. And then just some kind of recommendations, really, to wrap up, is that it's really important that we do think about opportunities for citizen science in zoos, and we consider how we can deliver those different opportunities beyond just, have you seen these things on site? We need to consider who all our users are and make sure that the underrepresented groups are also having an input as well. And then we need to think about actually how we can formally embed this into conservation education policies. I think I'm just out of time, but thank you all for, thanks for inviting me, and thank you for being here today. And then thanks to my sort of project team for the people that are behind that pilot study. And if anybody's got any questions, I'd be happy to take them.
Thank you very much, Ellen. Does anyone have any questions for her?
Hi, thank you. Very cool initiative. I'm going to ask you about your second aim. So you have them engaged now. Can you use the data?
Yes. Partially seems to be the answer. So what we looked at was, so we just, visitors, they did go a little bit rogue. So we asked them to look for 2 minutes, and some of them definitely did more than 2 minutes, and some of them definitely did less than 2 minutes. So I don't think it's reliable in terms of like proportion of time, but we did find that actually for some behaviours, at least the more common, you could look at kind of presence-absence of behaviours. So in terms of like, I don't know, maybe you could ask a question of like, is the animal engaging with this piece of enrichment? And if you've got like kind of a yes-no option, then you can start to build up, okay, well, this many visitors over this much time saw that happening. I think we need to go a bit further with it until we can be sure that people can actually reliably say, yeah, the giraffe spent 60% of their time feeding, and then we know that's actually relevant data. But at least if it's just telling us something about how the animals are spending their time, it's better than not knowing at all if we're too busy to capture the data. So, yeah.
That's great, very promising.
Yes, fingers crossed.
Hey, very cool. Do you think you could have, like, if it's, I assume it's all on their mobile phone, so they could, to get time data, you could just get them to say they've started recording, and then they can answer some questions, and then they press a button when they've stopped recording and they can no longer answer any questions, and then you know that it was during that time period. They didn't know what the questions were before, and they can't answer them after these 2 time periods.
Yeah, that would be a really good idea. So it would be nice, I was actually just saying to colleagues the other day that it would be really nice to sort of develop like an app to have a little bit more control over it. So what we had was a survey, and the reason I know some people were definitely doing this out of hours is that like it would tell me they completed at 8 p.m. and the zoo shut at like 5. So they definitely didn't know what the giraffe were doing at 8 p.m. So it's just kind of, but the other thing we had was like, they just spent longer than 2 minutes actually at the enclosure, but maybe if we do look at having like a timer, or having like a time, I was just saying to Maria earlier actually, maybe having like a tablet or something on the side where, you know, like when you go to the toilet, it's like, did you enjoy this experience? Happy face, sad face, and have a tablet that's like, can you see a giraffe? What's it doing? That kind of thing, potentially. Yeah. Yeah. Or I saw them be like, yeah, I'll do that. Scan the thing and then walk off. And I'm like, okay, thank you. Like, so, yeah.
I have an online question from Saskia Verbruggen. The question is, was the data gathered useful? Did you check already?
Yeah, so we did check. I think it was useful in so much as it told us a lot about, so this was obviously only a pilot study, so it told us a lot about like what people are thinking of in zoos, which is really useful for us as educators. But in terms of management of animals and can we use that data, potentially. I think we could possibly do more with the affective state stuff. So I've got a PhD student at the moment who is just doing some work developing like generalized qualitative behavioural assessments to try and use those with visitors. And I think that might generate a bit more information about like the affective state, not necessarily the activity budgets, but just perceptions of welfare from that angle. Yeah.
We're just going to wait for a few seconds. John Taylor had mentioned he had a question for you. John, would you like to ask your question to Ellen?
If I can, yeah, how do you feel here on it? No.
No pressure.
What have we been doing? I didn't hear the end of your presentation.
It was great.
I'm sure it was. The rest of it was brilliant, I have no doubt. It was half comment, half question, really. The question is, how do you come back on the comment? Which was, you went from the Living Planet Index, yes, to here's all the threats on biodiversity, yes. We must engage the public more, yes, I'm with you, to collect more data. Hang on a minute. I wasn't aware that a lack of data was our biggest problem. And I guess my comment is, do I agree with your threat analysis? Absolutely. Do I think that we should be engaging the public? Absolutely. Should our main focus on engaging the public be getting them to collect data for us? Is that the biggest problem? Because that's what would worry me, is people go away thinking, oh yeah, we've really got to engage the public, so we can give them an app and they can go and collect more data, rather than perhaps trade in their large camper van for a bicycle and those kind of things.
Yeah.
So that was I lost the logic there.
Yeah. So I think I would not say that collecting data is like well, not having the data is not the biggest threat. There are many other threats, but if you can, the logic behind them collecting data is that them collecting data on an animal's behaviour encourages them to think more about that animal, that encourages them to feel empathy for the animal, that encourages them to kind of potentially go away and do more pro-conservation behaviours. And so if you then embedded this with, we didn't try to put any kind of conservation messaging in here, we just were like, tell us what this animal's doing, how do you feel about its welfare, what does welfare mean to you, all that sort of thing, from an engagement point of view. But if you could slot stuff in, like, you know, okay, thanks so much for submitting that data, you know, did you know that like giraffe in the wild are experiencing X, Y, Z? And you can start to put conservation messaging in there. And then like, this is how you can make a difference. And there's obviously a whole host of research that shows that if people make a pledge to like out loud to someone, they're much more likely to then follow through on that pledge. So there's definitely areas you can go like after this. And I think that actually the whole process of citizen science is important from a point of view of getting people to care and then getting people to recognize that like actually these animals are really important. Like we already chose species that it's probably quite easy to like and to feel empathy for, but actually we can maybe sort of start to think about doing that with a whole range of other species as well that people maybe have thought less about to kind of engage that point of view. I don't know if that answers your point.
Yeah, so this is it's citizen science and engagement with the eventual aim of raising awareness of conservation issues and the species involved, rather than for its own sake.
Oh yeah, absolutely. Yeah, I mean, like it's also great from a zoo point of view if we can collect data on what our animals are doing and we can use that data to tell us more about how to keep those animals, and then that frees up keeper time to go and do other things and maybe other conservation messaging. So yeah, it's all part of a bigger picture.
Thank you very much, Ellen. Another round of applause, please. Okay, so our next talk is with Dr. Paul Rose. Here he is. He is a lecturer in psychology with a focus on zoo biology and animal welfare. His research explores animal management, behavior, and the societal role of zoos. He also contributes to policy and guidelines related to zoo management and conservation. So, Paul, whenever you're ready.
Thanks very much, Idu. Hi, folks. Thank you very much for the invitation to speak. I've got a rather grand title to this talk, but basically this boils down to, do flamingos have friends? And if so, are they the same as people? So I want to talk a bit about social network analysis, its relevance to animal welfare, and how we can communicate those stories to our visitors. So I want to just briefly introduce the concept of social network analysis to those of you that have never done this before. We've had a couple of talks already from a conservation perspective as to why this is relevant. So I'm just going to go over the nuts and bolts of what that means. I'd like to talk about how social network analysis can inform zoo animal management, and then think about how we could then use that to engage with our visitors in a way that links their lives and their social experiences with those of the animals that we see when they go and visit the zoo. So if we think about a One Health paradigm where we've got links between the human, the animal, and the environment, and that can impact on health and welfare, we can kind of use this as a framework for understanding why sociality leverages so much impact over animal health and animal welfare because of all these interconnected links. So we know that sociality is fundamental to good health and welfare for social species and to some extent solitary species if we get that social choice wrong. We are aware that many animals that are social are highly complex in their behavioural choices, and the restrictions placed on them from, regardless of how modern your facility is, but the restrictions placed on them by simply being managed can reduce their social choices. I think Maria's talk earlier beautifully illustrated that with some of the challenges of breeding mink in captivity. So we need to look at moving beyond just description of, oh, these animals are seen in a group, these ones are solitary, these ones live in groups of 10, these ones live in groups of 20 or whatever, to understanding the individual's impacts on that social group and also the social group's impacts on that individuals and how that might change at different times of the year. So, for example, the species that I will bore you with the most today, which is the flamingo, has this really kind of flamboyant array of social behaviours that changes with its physiological state and changes temporally and with season. So that could be something that's quite easy to manipulate in captivity if we have enough individuals in our groups. But other species, like these rather fabulous hornbills at the bottom, that flock together at certain times of their developmental period, when they're most plastic, i.e. when they're youngsters and they've just fledged and they're looking for a partner, the social information they gather in that flock is critical to pair bonding and future reproductive success. So is that why big hornbills are really challenging to breed in zoos? Because we don't allow them this early developmental stage and flocking behaviour. So do we have a mismatch for some species between social choices, their wild ecology, and what we're able to provide in the zoo? So one of the things we can do with social network analysis is to get an insight into individual choices and group level effects and marry those up together. If you'd like to know more about any of the social network concepts that I'm going to talk about today, there's a whole host of literature out there. I'm not tech savvy enough to do a QR code, so you're going to have to get your camera phones out or scribble stuff. So apologies for being a bit of a dinosaur. There'll be lots of references that you can't read and will flash up very quickly, so come and shout at me later. But basically, social networks and inputting those data tell us a lot about welfare metrics at the species-specific and group level. They give us cues on population management, such as when to move individuals around at natural dispersal rates or ages, for example. They give us an idea of whether or not conservation is going to be successful based on the recruitment into a population from breeding partners. We're able to manage behaviour by looking at flashpoints of aggression, for example, around particular resources at certain times of the day. And then we can change the design of our exhibits and enclosures with a database view on what are animals engaging with, what are they not engaging with, and what do they need. And all of these things kind of link back to keeping records, which we're told to do more and more and more in a detailed fashion in zoos and aquariums, to understand what is this population being managed at a particular place and time. And then those records can travel with those individual animals if they ever move to another facility. So SNA is basically a connection of information on the animals themselves, and in this schematic diagram you can see on the slide, the animals are the circles. We call those nodes in posh science speak, but that basically means the individuals themselves, and you can see all the lines between them. The connections are called edges, and we had some lovely examples of that yesterday from the giraffe talks with the nodes and edges and the thickness of those lines between different giraffe in different herds, indicating the strength of the relationship between them. So what's really useful about SNA is that it goes beyond just looking at the group, as I said, to understanding the role of the individuals and how they impact on what the group can do. We can then make evidence-based decisions because we are aware of these stronger interactions. There might be some kind of adaptive or fitness benefit to that connection, so we need to make sure that's preserved or maintained in population management. We can also think about prediction of particular outcomes, and they might be husbandry-based. So if we give enrichment or if we change enclosures or something like that that impacts on what animals can do, we might know that this individual is going to dominate, and these are the ones that are connected to that individual that might benefit, or these are the ones that are not connected and they might be at a disadvantage. So again, the information you put into the network gets you a lot out of it on how animals are responding to their environment, both socially and physically. And we can also layer in welfare measures alongside of our connections of dots and lines to see whether or not different metrics that we've used to understand welfare, for example, things that link to physical health or things that link to psychological health, impact on the structure of a network at particular times of the day, month, year, season, whatever. And there's a paper that I wrote with my then PhD supervisor, Professor Darren Croft, who I have to give all of the credit for to all of this social network stuff because he has literally written the book on it, which basically goes through the key elements of zoo animal management and how SNA has a relevance to that section of zoo management. And that's an open access paper in Animal Welfare, if anyone's interested. Terrifying that that's over 10 years old already, but never mind. So imagine that you are looking after a social species, and imagine that you are in charge of the environment it lives in and the choices that it has in this environment. Your individual is going to experience a group-level phenomena, which means there are substructures and smaller cliques around that individual that will impact what it can do. And that ultimately is going to affect the size and scale of the population, because those social choices will come from the number of individuals that are available. And likewise, the social group is going to be dependent on everybody else that's around it. That's why analysing data on social behaviour can be challenging, because these data points are not independent. Your social choices are impacted by the friends, family, colleagues, relationships that you have around you. And we need to bear in mind that lack of independence, and that's why social networks give us a clue. So let's meet one of the animals in this flock. This is Fiona. Fiona is friends with Fred, Felicity, and Frank. And this is our little flock of flamingos that we like to see together. I then ran out of names that began with F for flamingos, so I then stopped. And if you imagine that you're really interested in Fiona's social choices, and you are managing this population, and you want to know why those animals are together, you can measure them over time, you can look at inferred associations or direct interactions, and that gives you a clue as to why they've grouped together. And we can see these bonds are maintained within a wider social group, and then when we look more detailed into the flock, we also see there are other quartets and trios and small groups of flamingos that are also choosing these preferential associations. So this network is built up from the individual outwards, but also from the network overall, collapsing in on the individuals. So it's this two-way flow of information that's really useful to understanding what is it about the network that we can adapt to manipulate or change to maybe promote positive behaviours or promote positive welfare outcomes. You can't read this slide, but basically what this shows is just a range of examples of literature on social species in zoos, notably the flamingo, that show these patterns and trends in time and space across facilities, species, and flock sizes. So there's a couple of examples in here. They're all open access if you'd like to look at them, that give you an idea that regardless of how you manage your flamingos, regardless of the number of birds in your flock, and regardless of where your facility is, these social choices that I rather facetiously explained with Frank, Felicity, Fiona, and whatever the name of the other one was, on the other slide, they hold true. So you can have 300 flamingos or you can have 10. And there are preferential associations within those flocks. And there must be some kind of biological meaning to that. That social choice must be important to those birds. So we need to try and preserve that in our animal management actions. If we look a bit more deeply into a network, so this is from a paper in Applied Animal Behaviour Science, which just shows 1 year's worth of data across a range of different flamingo flocks. And this is just a network from one flock. Incredibly traditionally, I do apologise, boys are in blue and girls are in red. And you can see the connections between the animals in the filtered network, which is here, and the unfiltered network with all the connections, which is here. So by filtering and removing noise from that network, we can clearly see that there are really, really strong bonds that exist between birds. And then if we were going to move flamingos between facilities, we could identify, well, if we were going to move this one, we also have to move that one. And note that these are not just sex-typical bonds for reproduction. These are not just male-female pairs. These are pairs between males and females. So the bonds themselves are not just for the purposes of reproduction. The bonds are there because, to be really anthropomorphic, they like each other. They are deliberately choosing to spend their time with other birds in that group. And the largest flock of birds I looked at was just shy of 300. And this gives you an idea of the complex nature of a flamingo flock. So what this network shows you is structured by behaviour. So we've got birds that are in blue that have been doing their courtship display. We've got birds that are in yellow that have been seen to do the nesting behaviour. And we've got birds in red that have been seen to do both. So if there are birds that are displaying but then not nesting, or if there are birds that have nested but we've not seen their courtship display because maybe they've got these long-term partnerships and they're not getting involved in wider group activities, is that having an impact on reproductive rates? So we can then look at tweaking or changing the enclosure, perhaps mixing up the flock dynamics slightly to encourage those birds in blue that are displaying but never finding a partner to eventually have more social connections that might lead them to eventual reproduction. And there's some really fascinating research out there that shows if you can manipulate and manage your flamingo flock at non-breeding times of the year, perhaps you've got a large flock and you can split it in half and then you bring them back together, you can make wider ranges of connections that then positively impact on nesting. One of the things that's slightly challenging with social network analysis, but I would encourage you to have a go and try if you're interested, are the choices of stats that are available. We have to do things like Mantel testing or permutation tests. This is not a statistics lecture, please don't run. But they remove the challenge of non-independence. So you are able to do some forms of tests that allow you to deal with the fact that all of these data points are connected to each other. And some of the literature, as I mentioned, by Professor Darren Croft, goes into that in much more detail. So just to kind of summarize that before I finish on how do we explain this to the wider world, I hope I've shown you in that very brief run-through that flamingos have complex societies. These are not just random collections of individuals together, and we need to conserve that complexity in animal management regimes. We've got enduring bonds within our birds, so one of my papers builds on that original network that I showed you, but 5 years later, and the same consistency in these friendship bonds still exists. And we know that there are environmental factors that impact on the friendships that flamingos have. So these can be within the breeding season and outside of the breeding season. So it's not just a bonding for reproduction, it's a social bond that is persistent over time, and that must be something down to choice of individuals that those birds really want to invest their time in. And if we consider the context behind the network, we can then look at things like social support or buffering in aggressive interactions or things like that. Because ultimately, if we have birds that are more peripheral to the flock, for example, youngsters with fewer social connections, are they then at a disadvantage because they're not getting access to the best or most favourable areas of the enclosure? I had a paper with a student of mine, Abby Loader, and we showed just that. Juvenile flamingos are very peripheral when they're not bright pink and beautiful looking to the rest of the flock. So we can use those data to change the enclosure setup and change husbandry to give them access to more favourable resources. So as Ellen beautifully explained in her last talk, we need to engage with our audiences, be they transient visitors to our zoos, people that come along very regularly, and those that are really engaged with wildlife and really, really like animals. So we need to remember that science is only good as the audience that it reaches, and we can use social network information to look at our visitors and to underpin our educational messages with a little bit of storytelling, a little bit of narration, a little bit of, dare I say, the soap opera that goes on in the kind of flocks, herd, social groups, whatever it is of that species that we're managing. So we can create a narrative, and a social network allows you to do that because you can pinpoint individuals. You should know them by their identifiers or names or whatever it is that you know your animals by, and you can link their behaviours and their choices to human friendships and to human social structures. We then get more impact because we can deepen our visitors' appreciation for these important stories that we're trying to tell with our zoo animals. We make science more accessible by linking it to things that people understand. And these individual characteristics in long-lived species, so my favourite throwaway fact about flamingos is they can live for about 80 years. That's a long time in the zoo, but a lot of stories you can tell with a species with a very similar lifespan to us as humans. So if you'd like to know more about that, because I have nothing better to do with my time, I write a diary of my work at the Wildfowl and Wetlands Trust, or WWT. It's WWT's Flamingo Diary. Just Google search it, you'll find it. If you are ever struggling to sleep over night time, this will cure it. And I just try to explain this social behaviour, the research that we do, to people that would like to know a bit more. The hook is them seeing the birds in the zoo. We then have various signage around that they can engage with a bit more and various artifacts about the animals. And then if they're super keen, they can go and have a bit more accessible journey into the research behind the animals themselves. So I wouldn't be able to do this without a social network's background of knowing who the birds are, what they're up to, and their different demographics that we can then tell to our visitors. And this links across all of the different species of flamingos. So the 3 common species at the top, which we often find in zoos, Greater, Caribbean, and Chilean. We can then use the stories of these birds to these rarer species that we are less likely to see in zoological collections. But because we are engaging our visitors with these species that they're familiar with, or hopefully more familiar with, we can use the same ideals to explain the challenges and conservation needs of the rarer species that might be many thousands of kilometres away in countries that our visitors have never been to. So my conclusion is social network analysis, in my incredibly biased view, is a very good measure for understanding the impacts of welfare at the individual and the group level. And we should try to incorporate that more into the welfare frameworks that we use. It's also super important for evidence-based management because it's a direct look into the individual social choices, and that then allows us to change what we're doing to accommodate all of the different needs of the species that we are managing. We can then explain zoo husbandry, be more transparent, like one of Edu's questions in yesterday's discussion panel. We can be more transparent by explaining why we have this number of flamingos, why they're in this group, and what are they doing with each other. And ultimately, that could lend us a better narrative to explain the role of those animals in the zoo. And hopefully, we can then extend the application of social network analysis, which currently is quite limited to charismatic mammals, and think about all of the other myriad of social species that we manage, where we can recognize individuals and we can ask these taxing questions on social choices and then the impact of those social choices on everybody in that group. So I'd just like to thank everybody that has been involved in the research that I've just presented, all of my BSc and MSc students that have helped collect data, everybody that manages the birds that has allowed me to annoy them and the animals themselves during data collection. And this collection of references here are just general ones on social behaviour and social networks with an applied perspective. So if anyone's thinking of doing this in the future, that reference this there might be useful to you. Thank you very much.
Thank you very much, Paul. We have time for one online question and one quick in-person question. The online question is, can zookeepers establish this social network easily, or do we need scientists if we want to use it for management decisions?
You can get social network data from photographs, so long as the photo is good enough quality. So zookeepers can take a photo of a social group at a snapshot. That can be uploaded to a central database, and then your researcher, research student, or whoever has been trained to pull the information out of the photo, that data can then be put into the fancy stats that I was talking about, and then the network can be presented to them. So it's a collaboration to get the network, but the actual collection of data is relatively simple.
Okay. Are there any in-person questions? Yeah.
Thank you so much for that talk. Very interesting. My question sort of connects to that one. When you are trying to establish the relationships, I find it surprising that you can establish them just from the photograph, because that would mean that just their mere proximity can basically explain their connections, but you had such a complex network, and then the bigger circles and the thicker lines, and I'm wondering, don't you need to observe maybe different types of behavior to explain those different social connections? Like, what are the things that you're looking for?
That is a very insightful question. Had I got the chance to do the whole conference on how to do social networks, I would make you all go outside and we would look at the difference between interactions, which we as humans are really good at, and associations, which are based on proximity. Mm-hmm. So I had to collect multiple years' worth of flamingo association data to see were I seeing the same birds consistently next to each other? And that was how I was able to build my network. If you are working with an animal that does stuff to each other, like primates that groom, your network can be collected much more quickly. But an animal that doesn't do that, so you're just assuming that spatial distance is your measure of friendship, you require months and months and months of repeats to make sure what you're seeing is true and not just random chance. So your method chosen depends on the species.
Okay, thank you.
Please, another round of applause for Paul. Thank you. All right, so our next presenter is Dr. Benjamin Schulz. He is a curator here at the Serengeti-Park Hodenhagen. He is a biologist specializing in reproductive technologies for wild and zoo animals. He develops methods such as artificial insemination and contraception to support population management and genetic diversity. His work combines conservation goals with animal welfare and practical training experience. So please welcome Dr. Benjamin Schulz.
Yeah, so in the next 20 minutes, everything is about sex. Of course, this fun topic has a serious background. And unfortunately, mankind has managed to bring thousands of species to the brink of extinction. Loss and destruction of habitat, or through poaching, and also pollution and overuse of natural resources. All these problems are progressing as we speak here, and in the light of the so-called sixth extinction, both zoos and international authorities have discovered the value of breeding programs to act as reserve populations and safe havens for those that are threatened the most. So over the past decades, zoos have become more organized, more connected, and more professional, and have redefined their purpose from amusement collections via live museums to active centers of species conservation. And at the same time, their efforts to breed species have also developed. While in the very beginning, new animals were still easily sourced from wild populations, the change in legislations has made it necessary for zoos to reproduce their own stocks. This, of course, in the beginning, was for many species also rather unsuccessful. It was chaotic in the beginning and lacking a common goal. But breeding programs today are getting more and more coordinated. They are targeted on genetic variety, social compatibilities, and as well animal welfare at the same time. And that's the most important thing, they are becoming more successful as we speak as well, making a more thorough control necessary of who breeds with who, and keeping local space as well as global population structures in mind. So, assisted reproduction. When speaking of assisted reproduction technologies, I think that most people probably first think about things like in vitro fertilization, embryo implantation, maybe even in surrogate mothers. While all these things have been attempted in animals as well, they play little to no role in practical applications because they are still too complicated and too complex to use in wild animal species. Artificial insemination, on the other hand, and even more, sperm collection and cryopreservation for future use in gene banking have become more and more important in breeding programs. AI, in this case it's not artificial intelligence, but artificial insemination, can solve a number of problems associated with animal transportation, for example, with the logistics that are involved in that. Because now, a simple vial of semen can introduce new genetics into a foreign group without the need of disrupting strong social bonds, like for example in certain kind of cetaceans, in great apes, or they can to avoid or they can avoid to introduce partners of species who are generally very difficult to socialize, like many solitary big cats, for example. Okay, this paper is strange. So here it can be beneficial to use AI to avoid destroying an harmonic group or injuring or killing animals when introductions are very risky. However, when it is about reproduction in zoo animals, people often neglect the one discipline that is most common in population management, and that is contraception, a tool that everyone needs. So, over the decades, scientific progress has brought many studbook programs to success. In some cases, though, they became too successful. And as breeding programs must look for genetic variety, while at the same time keep on breeding to avoid overaging of breeding animals, a certain number of surplus animals will always be produced. Now, in some species, culling and feeding surplus animals to predators is a solution, as it would happen also in the wild as well. But advanced veterinary care and husbandry has also led to a number of other species that are not considered prey animals to reproduce successfully, more successfully than in the wild in some places, where deaths of young or adolescent animals before they reach reproductive age is usually common in the wild populations. So naturally, the public, as well as authorities that control zoos, want zoos to breed responsibly and not produce too much surplus and kill it then. In the case of primates, elephants, dolphins, and other popular ambassador species, the acceptance of killing animals just because they don't fit in a breeding plan is very low. And this is good, because zoos are places made by people for people. They are part of the society, and therefore they cannot afford to go just against what the society thinks and feels morally. So zoos are not exclaves of the humane practices and ethics. So for these and other animals, a more gentle approach is necessary to prevent overbreeding and surplus animals to reproduce, and that is contraception. Contraception is coming in many forms and methods. However, these are far from perfect and bring other problems as well that zoos have to deal with, which is why it is always a careful act of balance for zoo managements to decide what happens with a single animal if the decision is to sacrifice its welfare for the good of the population. As examples here, we see that contraception can happen as a physical form. Physical form means separation of males and females to avoid breeding. It can happen in a surgical form, castration, sterilization, but all of this is, of course, a very invasive procedure. And it can happen in the form of chemical contraception, like implants, injections, or other kind of hormonal contraception forms. And now, in the following examples, I would like to give an overview of how complex this topic can be as every species has different needs in social and sexual behavior and comes with unique physiological features as well. And I would like to start with the Hamadryas baboon. So this species is representing all baboons and mainly also other primates. This one is not endangered, and it is reproducing so well in zoos that population management has difficulty to keep up with it. So why actually talk about them here? So yeah, as the techniques and methods that could make the difference for other highly endangered primates can be developed using this flourishing species help without trying things that are not tested on other more high-valued species. So Hamadryas baboons, like other baboons, are living in large mixed troops, where one or several dominant males can monopolize mating rights over the females. To do this, they use blunt force at times, and they are not real gentlemen with their females either. So if a female doesn't submit to males' advances, They can be bitten, beaten, or just pulled through the enclosure by an enraged, muscle-packed pasha. Mating is often consummated as a result of such threats and not a romantic affair at all in these species. The typical grooming periods that often prelude or accompany mating bouts cannot distract from the fact that copulation in this species is a male-centered behavior with little room to female consent at all. Interestingly, and not without looking at the human society, the higher up in the hierarchy a male baboon is, the more prone they are to just take what they want. While lower-ranked males need other, more gentle skills to convince females to mate. Or shortly, the more power a male has, the closer they are to be wife-beating jerks. Now, these baboons and others of their kind are widespread in zoos and have reproduced too well. And instead of sending animals on exchanges and promote breeding, the EPs of these species are busy preventing more offspring and somehow finding a path to put order into the chaos. As a close cousin, the Guinea baboon has reached, let's say, unwanted fame as the decision of a German zoo to cull healthy young animals and continue breeding sparked a public outcry and increased resistance by animal rights groups, and has even met some disagreement in the zoo world itself. So maybe the management has thought that these primates do not meet much sympathy, but does it make it right then to just shoot animals that have landed in the surplus category with their own fault, without their own fault? This is definitely a difficult question of modern ethics, and the first decision by the public was clearly that it is not so okay. So whatever the long-term outcome will be, zoos are forced to think how they want to deal with this problematic species in general. A coming solution might be coming with progress in reproduction sciences. A non-hormonal contraception method that is only working on males and distributed through their food might be the tool in the future that could enable zoos to finally handle this and other species, and at the same time providing the means to permanently or temporarily contracept even other endangered primates without disrupting their natural behaviors or destroying their fertility forever. So, yeah, in conclusion, this species here in zoos needs no help to reproduce. They need contraception at the moment that is easy to use and monitor. Let's go to the second example. That's the white rhino. And the white rhino is known as a stamina beast. These guys have suffered a lot under their reputation. The main reason for poaching rhinos and selling various parts is that they take their time when they are engaging. Copulation can last up to an hour. That might be a top result, but the average time still puts every human to shame. So during copulation, the male rhino also ejaculates multiple times. What started out as a real treat from evolution in regards to pleasurable experiences ended in merciless hunts. Humans have managed what no other natural disaster could do over millions of years to this species. They have made one of the rhino species extinct and bringing all others to the edge. So naturally, the problem with rhinos in zoos is definitely not contraception in this case, it is conception. With their slow reproductive rates and long interbirth intervals, rhinos are not quick ones to increase in numbers. And they never had to before humans popped into their lives. So their sheer size also demands a lot of space to keep them, and transporting adult rhinos is never a routine task to do either. But females have to become pregnant soon after maturing. Otherwise, they might never conceive successfully again. So if the right mate is not available, then AI must do the trick, at least to achieve the important first pregnancy. Here at Serengeti-Park, we currently are very lucky to have a formidable breeding group, but nevertheless, the problem in the population remains, And the secret to success is global cooperation and combined efforts to ensure this species' future. So in general, what the rhino needs is a lot of support to reproduce. Example number 3, also very popular species, the African lion. The only social big cat has a special place in reproduction physiology. Like all cats, also lionesses need the stimulus of mating to release their eggs to fertilization. As a result, they happen to just love sex. Females can actively seduce several males over the course of several days and exhaust all of them, demanding coitus from their partners every 15 minutes is essential for a successful impregnation. Like that, females in this species are in complete control of sexual interactions, and the male has nothing to say in it other than comply and do his job. So if he does it well enough and often enough, that's the most important thing, then she will dedicate her eggs to his sperm. Known to reproduction scientists, this is known to reproductive scientists as induced ovulation. And this feature alone does not create any trouble in other solitary living big cats like tigers or leopards regarding population management. But in lions, of all things, it is in fact their social lifestyle that creates a complex approach to population management. Because contrary to solitary big cats, lions can quickly reproduce so much that they explode the space capacity of any zoos holding them. Additionally, while in solitary big cats the separation of the sexes is just natural and helps controlling the speed of reproduction programs, this is not practicable in lions. It would disrupt their social groups and could make it really hard to reintroduce these partners later on, when you are ready to breed. So, logistics of contraception suggested that preventing pregnancies should be easy. In a group of just one male with several females, just contracept the male and you are out of trouble. But here, induced ovulation gets in the way. A contracepted or sterilized male will still proceed to serve his lionesses, which from the social aspect is a good thing, but physiologically, the repeated copulations that lions perform leads to waves after waves of hormonal peaks in the females, ovulating repeatedly, but no conception ever occurs. And in this case, I still have a lot, sorry. Lionesses frequently enter a so-called pseudo-pregnancy, and that can lead to other complications in the future like uterine inflammation, endometritis, and even tumor development. So shortly, while the males live on their lives almost normally, the females are suffering the consequences of contraception in lions. The only safe way to contracept lions are implants for females that shut off their whole reproductive cycles. But again, this has consequences for the social hierarchies and behavior, and the implants that are available are developed for use in domestic animals like dogs and cats. So implants that work well and reliable for the time of year in dogs can be excessively long in duration, up to 5 years, which, considering the average lifespan of lionesses and their reproductive age, often means a complete shutdown without reversion. Let's come to the last example. In this case, this is the South American sea lion. Similar name, but no, it's not the same problem. Pinnipeds are slowly and seasonally reproducing, unlike the frivolous lions who do it anytime, any place. Sea lions have a strict rule: once a year, same place, same time, that must be enough, at least for the females. Their copulatory patterns are clearly male-centered as well, as he's the only one who serves all his females and uses his superior weight and strength in case she's not 100% into it. All that doesn't matter to him. He mounts and he finishes the job, spends maybe 15 minutes with every female, and then he sees his job as completed. So this is the typical sex life of pinnipeds. But what makes it challenging for zoos to manage it? Well, first of all, a strictly seasonal mating single pups per female? That shouldn't be too difficult to get your hands on, or is it? Well, in modern EP-directed breeding programs, it is not about the numbers of pinnipeds being born, but about genetics. And the natural social structure of one male claiming a harem for himself has led to uncontested breeding in zoos, often over several seasons. And the result is genetically very close offspring over and over again. And too many males among them either, because naturally the offspring in mammals is 50-50. And that shouldn't be the case then when there's one dominant male over several females. All the available contraception products that we have, that we can use for pinnipeds, are not really effective and also not reliable, because the massive blubber just absorbs a lot of it, and naturally male sea lions experience a massive spike of testosterone secretion around the breeding season, which is almost non-shuttleable by anything chemically that is on the market. So the current problem persists, and interestingly, the same research that could in the future be a solution for the exploding baboon populations could also help contracept sea lions in the future. Let's come to the conclusion. To breed or not to breed? This is the question here. But this is no longer a simple question for zoos to ask themselves, but it is a complex field which demands global cooperation, careful planning, and the expertise of animal trainers, veterinarians, population biologists, genetic, physiological, and pharmaceutical scientists, to improve and give us tools that help us do what modern zoos are obliged to do. We have to manage our animal collections while retaining good animal welfare for individuals and groups alike, retain a good physical and mental health for the animals in our care, and all with the goal in mind that we want to keep populations that are viable, young, fertile, and genetically diverse at the same time. Because what we have learned from the past is that our decisions today might be very well relevant in 50 years or more from now on. So thank you for your attention, and I'm happy to answer any questions or comments right now.
Thank you very much, Benjamin. We have time for maybe one question. Jessica?
Thank you for that. It's very interesting. So I had multiple questions, but I'm going to narrow it down since we only have time for one.
Okay.
The food-based contraception, has it already been implemented with the baboons? Is it successful? What stage is that at? And how many different species can it be used or tested on?
This is actually a human medicine product that is right now in clinical studies with humans. And we are right now working in the EASA as well as the ASA to get hands on this product and start a trial phase with some selected zoo animal species.
Okay. So it's very new.
It's very new. It's a non-hormonal contraception, and it's the so-called pill for men that is now on the market in the US and the UK for now.
Okay.
Let's hope it's coming to Europe after some time as well.
Thank you.
Thank you too.
So if anyone has any more questions for Benjamin, please just ask him individually. Sorry to shorten it for you, but thank you so much. Great presentation. So another round of applause. Thanks, Benjamin. Hello, hello. Okay, so our next presenter is Beatriz
Guimarães.
See?
You don't read the U.
Guimarães. Okay, so Beatriz was one of our Erasmus Plus program students, and she completed her master's thesis project here. She's an early career researcher in wildlife management and animal behavior. Her work focuses on how enclosure design affects animal behavior and welfare in zoos. So she aims to apply scientific findings to improve animal care and conservation practices. So Beatriz, it's all for you.
let's see if I can do it. So, hello, my name is Beatriz Guimarães, and today I'm here to present my work on how captive pygmy hippos use space and time in a mixed species exhibit, and what this can tell us about their behavioral plasticity, welfare, and enclosure management in a zoo. So, pygmy hippos are an endangered, elusive, solitary, and semi-aquatic African ungulate native to the forests of West Africa. And studying these species in the wild is extremely challenging due to its mostly nocturnal behavior, dense forest habitat, fragmented distribution, and even the tendency to avoid other mammal species. So because of this, the research is quite limited, even in captivity. So, and given the rapid decline of the wild populations, behavioral research under managed care still remains quite important for us to provide management and conservation planning. So the aim of this study was to evaluate how two captive pygmy hippos organize their space and time in a mixed species exhibit, and more specifically, I quantified their activity budgets both indoors and outdoors across day and nighttime. I also characterized sleep patterns and resting proximity between the pair, analyzed space use, and mapped where specific behaviors occur to identify functional hotspots. So the study was conducted here in the West Africa section, and this is a mixed species exhibit that houses pygmy hippos alongside other African ungulates, as well as green monkeys. The primary animals involved in this study were a male pygmy hippo named Michelin and a female named Zala, and they were kept in an indoor facility that had an adjacent outdoor area with grassy and open regions, and also wooded, a slope, pools, and as well as a monkey island. The data were collected from mid-April to late June 2025, and the study was divided into 7 distinct phases to reflect the changes in the ungulates group's composition. And a key event during the study was the gradual introduction of the female into the outdoor environment with the male, as prior to this she only had a restricted outdoor area and no direct contact with the male. Therefore, this study allows the assessment of the behavioral response to both environmental as well as social changes. So behavioral data were collected using scan sampling during the day, both indoors and outdoors, and at nighttime we complemented the scan sampling with 3 hours of continuous observations to capture more detailed nocturnal activity. Behaviors were defined using a detailed ethogram, and outdoor observations were recorded alongside environmental factors such as weather conditions, temperatures, and the number of cars. So for the simple analysis, we observed and combined consecutive sleep observations into bouts, allowing for the comparison across day and nighttime and indoor and outdoor settings. The space use analysis was done by using QGIS-based mapping. Kernel density estimators were used to identify core use areas, and the spread of precipitation index allowed us to assess how evenly or unevenly the enclosure was used. Together, these metrics helped us understand which areas were preferred and avoided in this enclosure. So regarding the results, and I know it's a lot, so we just compiled what we could. So regarding the indoor daytime activity, the male spent most of his time resting, whereas the female dedicated a larger portion of her time feeding. We also observed a clear difference in their resting levels, underscoring the differences in energy allocation even when both individuals were in the exact same environmental conditions. Regarding the nighttime, both individuals displayed very high levels of resting, and this is in contrast to the species' natural nocturnal activity in the wild, which highlights their behavioral plasticity, showing that they still were able to adapt to this captive routine while maintaining their ecological requirements. We also observed some behavioral variation across the study, with the female showing differences between feeding and resting between phases D and G in both cases. And these changes may be a response to environmental changes in the enclosure itself, as well as natural adjustments related to the female's physiological cycle. So this analysis revealed a clear distinction between daytime and nighttime allocation, with high activity levels during the day and a markedly reduced activity at nighttime. Regarding the outdoor, both individuals spend nearly half of their time feeding, And this is particularly interesting in a zoo context as it coincides with visitor presence. Therefore, we have more visibility opportunities for the visitors while still allowing for the animals to control their use of space. So regarding the environmental factors, the number of cars had a significant positive correlation with resting, social contact, and resting together, indicating that during busier periods of traffic, pygmy hippos tend to come into closer contact with another individual. Regarding with temperature, with higher temperatures, pygmy hippos tend to engage in more swimming. So as temperatures rises, pygmy hippos tend to engage in more aquatic activities for thermoregulation. Now moving on to the sleep analysis. Both the daytime indoors and outdoors sleep outs typically lasted around tens of minutes, while at nighttime this increased for 3 hours. This once again highlights this difference in sleep patterns, highlights the shift towards a more diurnal lifestyle suited to their captive setting. Regardless of the time of the day, both individuals displayed a specific preference for resting sites. In particular, inside, the male preferred to rest near the corridor's gate, and the female preferred to rest in the, between the corners, between the corridor's gate and the gate dividing each box. Thanks to these findings, the park's management has already been able to adapt the placement of their bedding to adjust to these preferred sleeping locations, meaning that this study had already a direct and positive impact on their daily welfare. Regardless of this, outdoors, both individuals have the same preference and resting on the Monkey Island, where they were not disturbed by an antelope species. So space use was clearly non-random, with pygmy hippos preferring to rest, concentrating their activity in particular core areas, such as Monkey Island. The SPI values also back up this information, indicating an uneven use of the enclosure. So there was a preference for particular functional zones rather than homogeneous exploration of all available space. Regarding environmental variables, we didn't detect very strong relationships between it and space use. So the clear shift towards decreased activity shows that our pygmy hippos displayed behavioral plasticity while still being able to adapt their captive routine to a, management setting. So the strong use of water bodies and sheltered refuges highlights once again that pygmy hippos were able to constantly prioritize their key ecological requirements while still shifting toward a more diurnal lifestyle. The clear individual differences in activity budget, sleep behavior, and resting sleeping sites underscores that the behavioral responses were not uniform, and therefore welfare assessments at an individual level should be prioritized in mixed species exhibits. And the tendency for the pair to rest closer together during busier traffic periods and their shared use of undisturbed areas such as the Monkey Island shows towards an increased social tolerance, as well as reinforces the importance of sheltered areas within a complex enclosure for this species. So this study had limitations. In particular, we only had 2 individuals to study. We also had a relatively short study period with no seasonality, as well as some visibility constraints due to the enclosure size. And the lack of physiological measures meant that this study was solely based on behavioral and spatial data. Nevertheless, it still provides very important insights in the welfare assessment in zoos. So the captive pygmy hippos in this mixed species exhibit displayed behavioral plasticity while still maintaining core species typical traits such as the strong reliance on water bodies and shelter refuges. Environmental complexity and access to spatial choice allowed them to constantly prioritize key ecological requirements even as they adjusted to the captive settings, highlighting positive welfare outcomes. And also by combining behavioral activity budgets, sleep analysis, and space use mapping, we can provide a non-invasive practical framework for welfare assessment and evidence-based enclosure design in zoos. So it is important for captive pygmy hippos exhibits to prioritize easy access to pools, shaded and quiet refuges, as well as provide structurally varied areas for the animals to avoid other individuals when needed. By using flexible feeding locations and enrichment, we can broaden the space use, while routine behavioral and spatial monitoring can allow us to adjust husbandry routines and social management over time to support positive welfare. And that's it for me. Thank you, and I look forward to your questions.
Thanks, Beatriz. That was a really good presentation. I am Bast, obviously. Yes, Paul. Paul has a question.
Thanks very much, and well done for trying to unpick something so challenging. Have you got any recommendations that could be used to improve the indoor housing to encourage nocturnal behaviours based on your data?
Okay, so you mean nocturnal behaviors. Okay. I think it's not about the enclosure itself. They do have stuff and like enrichment to provide the activity during the nighttime, but just because they are like put outdoors during daytime and they are active quite a lot, they naturally need to rest a lot during the nighttime. So it's not a lack of enrichment or opportunities to be active, is their lack of will, more like it. So they completely readjusted their routines to just have a very long nighttime rest. Welcome.
Are there any other questions for Beatriz?
So are they closed indoors at night?
Yeah, they always go, depends on the season, but yeah, they about 6 p.m. at least during summer, they go onto the indoor enclosures where they are completely separate. They don't stay together all night long.
Okay, and is that for safety reasons or what?
Yeah, I mean, if you ever see a pygmy hippo try to mate or come into closer contact with another, it's terrifying at the beginning, to put it lightly. They're quite aggressive. Well, not in a sense that they're gonna mangle each other, but for us as humans, you'll still see blood and cuts. So they still get along, but it's not safe to keep them into such a close enclosure together, because they are, in the end of the day, solitary species. Even though they tolerate each other in particular times and space, they still need to be away from each other. So having them together at nighttime could result in very bad things.
Okay.
Hope that answers.
Okay, there are no online do you have a question, Ellen? Okay, there are no online questions. Any other questions in person? All right, so one more round for Beatriz. Thank you.
Thank you.
Okay, so I will be introducing the second pair of the duo. It's Sara Valente. She is trained in wildlife management with a focus on animal behavior. Her research examines vocal communication and behavior in pygmy hippos in zoo environments. Her work contributes to improving enclosure design, social management, and overall animal welfare. So, welcome, Sara.
Hello, everyone. So, as you heard, I am the other half of this project, and I am very excited to be here today to share with you all the work we did last year. So because you just heard the lecture and it's on the same species, I will skip my first slide, which is on the general introduction about the pygmy hippos. And therefore, I will be starting on why we should study them in zoos. So basically, one first very important point to why we should study them in zoos includes the fact that they are very, very rare species, very shy, and obviously they had to choose a very dense environment to live on. And therefore, good luck trying to find one in the wild to study. The second reason why you should study them in zoos is because they are extremely sensitive to environmental changes, and therefore, if we are able to study them in a more controlled conditions, like the zoo enclosures provide, it allows us to understand how they respond and allows us to evolve and design better enclosures that will fit their needs better. Oh, I forgot my last. Sorry. Lastly, it is very important to keep studying them even in zoos because we have a big research gap regarding their ecology, their behavior, and also their vocalizations, which is what my study focuses on. And now, yes, let's go to my research goals. So with this study, I aim to investigate their social interactions and where they occurred on the enclosure. I also try to analyze their proximity patterns, to build their vocal repertoire, and lastly, to provide provide evidence-based recommendations. Moving on to the methods. So, as you heard before, the study was conducted here at the Serengeti-Park. Our main characters were two juvenile pygmy hippos, a female named Zala and a male named Michelin. And they live in a very complex mixed species exhibit together with, so it's the bongos, the Vlasvoks, the Sitatungas, the kudos, and the green monkeys. To collect our behavioral data, like Bia said, we did a 10-minute scan sampling method. However, I did collect a bit of different things than she did. So not only I collected the location of the pygmy hippo, but I also collected everyone else's locations in the enclosure, which was fun. I then had to analyze if they were interacting with other species. And also, lastly, I had another component to my study, which was the acoustic data collection. And so we did this from May to June, opportunistically alongside with the behavioral data collection. And we also included some overnight sessions. And we did this by using a handheld recorder attached to a shotgun microphone covered by a windshield to try to minimize as much as possible the wind and the background noise of a safari park. For our data analysis, so basically for social dynamics, we did negative binomial models as well as association testing. For our space use, we used GPS-based heat maps on QGIS. And lastly, for our data, for our calls, we analyzed them on Raven Pro. And then we did a PCA to separate them statistically and also perform some association testing to try to understand if there was a link between calls and behavior. Moving on to our results. So starting with social interactions, we had a total of 201 interactions during the study period. And even though the most common behavior, individual behavior was tusk display, the majority of events were actually affiliative. Moving on to our social interactions, we also saw it being skewed towards interspecific interactions, covering over 90% of our recorded events. So they were mostly just interacting with each other. For our spatial use, as you can see here on the heat map, it was definitely not uniform. So basically, we saw a majority of events, neutral events occurring around the water body, and a big portion of agonistic events occurring on the monkey island. For our regression models, we saw that weather conditions, when it was sunnier, it increased the number of interactions by 2.4 times when compared to other weather conditions. And conversely, our number of cars actually have quite of a suppressing, mild but suppressing effect on our interactions. So the more cars, the less interactions we saw. Moving on to our proximity analysis. So the pygmy hippos were found to have a smaller distance between the green monkeys when compared to other ungulates. And lastly, for our acoustic analysis, we saw, I hope this plays. Basically, we found 3 types of calls. The first one is the snort, the second one is the grunt, and both of these are post calls. And then we had one tonal call, which was the whining. So let's see if it plays. Do we have sound? Oh no. Oh, yeah. Okay. I hope you heard that. Yes, I know what it sounds like, but I promise it was a call, okay? This was the snort. The second one is the grunt. Yeah. That kind of sounds like a horse, in my opinion. And the last one is the whining. This one was quite annoying. Okay. So our PCA shows that these calls do form distinct clusters. However, and this is based on bandwidth and duration. However, the grunts, we need to take them with a pinch of salt because we had a very, very small sample size of them, and therefore they're here more as a representation aid than to anything else. And regarding our environmental context, what we found was that there was a clear link between certain behaviors and calls. So, for example, the whining was extremely related with affiliative or contacting behavior, but as well with separation, anxiety, kind of. Then we had the snorts, which were highly linked with mating or courtship behaviors. And lastly, our grunts that were more used in general annoyance, disturbance of their own environments. Moving on to our discussion. So first of all, and the one that I like the most, is that we found that they actually have quite a high tolerance, social plasticity, let's say. So they were able to tolerate each other quite well. And these already had a big impact on their management because they were previously housed separately on the outside as well as on the inside. But now they are able to share the outside enclosure freely without any barriers, which makes them one of the few pairs in the world that are actually to live like this. For our space use, what we saw, no, not space use, this is the, yeah, okay, sorry. So basically we saw that, space use was not uniform, it was actually quite functional. So the water bodies is where we found our most neutral behaviors, and this mainly included swimming because they could just swim around without minimal social tension, which was great. However, the monkey island was where we saw most aggression towards each other, and this can be explained by the fact that the monkey island is seen as kind of like a premium spot. So in the monkey island they have isolation, so they don't get bothered by other antelopes or other cars or people. Whatever, they feel pretty safe in there. But also, they're very sneaky. They just enjoy the snacks the monkeys sometimes drop. So food droppings are also quite a good incentive for them to stay in the island. Therefore, even this comes to show that even in a very successful mixed species enclosure, these kind of high value zones will still create some social friction, which I find it quite natural. Oh, and also, very important, they were not fighting for survival, just for preference. For our environmental predictors, what we saw here was that the sunny weather actually seemed to increase the number of interactions, and this can be easily explained by the fact that with warmer weather, there is more movement in and out of water, and therefore the hippos actually have more opportunities to encounter each other and interact. On the other side, the cars seem to have kind of a suppressing effect on sociality, and this is also quite easily explained by the fact that maybe humans act as a mild stressor to the hippos, and therefore they prioritize vigilance over socializing. And this is also a pattern seen in other zoos with other mammals as well. For their proximity, also quite funny story. Even though they were quite close to the monkeys, I find it very funny that the monkeys actually try to avoid the pygmy hippos at all costs by staying higher up on the branches. And for the ungulates, the other ungulates, they actually had a bigger distance from the pygmy hippos. It doesn't mean that they didn't like each other. I believe it means that they just use the habitat quite differently because the ungulates usually prefer more open and drier areas, while the pygmy hippos like shaded and semi-aquatic zones. And therefore, what we see here likely reflects ecological niche and not social preference. For their vocalizations, very, very cool because we actually found a very good relationship between calls and behaviors, and also we saw that they were actually using communication as a tool to kind of manage their own social boundaries and their territories, and so it was a very important tool for them to communicate with each other. Obviously, even though we had quite a few cool findings, there are limitations to every study. So our first limitation methodologically was because we only had a pair over a 2.5-month period, and therefore it makes it a bit hard to generalize to other age groups as well as other seasons or long-term patterns. Moreover, the fact that we used 2D mapping didn't allow us to statistically account for the monkeys avoiding the pygmy hippos vertically. Also, because we have, even though we use the shotgun microphone and we use the windshield protection, we still have a lot of background noise, as you could hear on the audios, and therefore they reduced our detectability, which resulted in a very small sample size of grunts, and I mean, who knows what else. And lastly, we need to take into account that these individuals live in a very unique exhibit, and therefore these results are very enclosure specific. Nevertheless, from a management point of view, it was still very, very important because it shows that enclosure design is one of the main tools we have today to mitigate conflict in a mixed species like this one. So very important to keep them with easy access to water pool, obviously, shaded vegetation, shaded zones and vegetation to allow them to regulate themselves and to build their own social boundaries, even in a more natural way, even in a very complex mixed species exhibit. Moreover, the acoustic analysis, also can be used as a very important tool in management facilities because it allows us to kind of anticipate social stress and as well separation anxiety issues even before they actually physically occur. Therefore, looking ahead, I believe it is extremely important that we continue this research in a multi-institutional and also longitudinal level, especially with a special focus on mother-calf dynamics, because we want our protocols to evolve within every single life stage of these individuals. And that's it. Thank you very much.
Thanks a lot, Sarah. That was really cool. Does anyone have any questions for her? Oh, no. Yes, Rick? Oh.
So first of all, thank you for your talk. It was very thorough. I had a question regarding the vocalizations, yes, because a lot of animals use it to communicate also when they cannot see each other. Did you also notice this vocal communication when the animals were out of each other's sight range or smell range?
Okay, so we did observe once, I believe, that there was a moment where Zala was still, because Bia explained to you that Zala was before isolated from the male. He had an electric fence, she could see him, but she was alone. And we heard her once doing a grunt because she touched the electric fence and she got hurt by her, she got a little hurt. And that was, I think, the only time I saw her doing a vocalization on her own, because the most common call we heard was the snorts. That's why I'm very, very sure there was a call. And they were mainly doing it when they were like interacting with each other, or sometimes it would happen when they would interact with other species, kind of as a complaint to like them to just back off, you know. But yeah, when they were apart and trying to communicate. Oh yeah, wait, sorry, I forgot the whining. The whining was used. It was mainly used when Zala couldn't see him, so she was the one doing the whining the most often, and she was kind of calling for him or for the keeper. I'm not entirely sure.
Okay, thank you.
I hope that answers the question.
Yeah, thank you. Really great talk. I just wondered if you had any thoughts on how to overcome the increased aggression on Monkey Island. So I know you said it's about resources, but does that mean that one then is not getting enough? And have you got thoughts on what we could do?
Oh no, I believe they're just princes. So basically, I think, it was quite interesting to see that because we were able to see before Zala and after Zala. So before Zala, there wasn't obviously any conflict. The male would spend a lot of time in the island, but I believe that he started spending more once Zala came outside, because it was really funny because they immediately liked the island more and they wanted to kind of fight for it as in a territory. But sometimes it would be a bit hard to understand if they were actually fighting for the territory, or if they were actually trying to, like Michelin was very interested in Zala, trying to mate with her, and she was just not having it. So it was kind of difficult to understand why they were actually fighting sometimes. But yeah, I don't know if there's anything to do to actually mitigate that kind of conflict, because it's just for preference, not because they don't have enough resources.
Okay, I have an online question for you. How do you plan to communicate the results of the pygmy hippo studies to the park's guests and the general public?
So I have been very grateful to actually work here in the park, and everyone was extremely welcoming. And we are trying to eventually find a way to publish. So speaking to the park about the results was not very hard because the park was very involved. But yeah, speaking to the public, we're trying to eventually organize this in a paper to show our results.
Thank you, Sarah. Another round of applause, please. Okay, and we have our last in-person presentation from Dr. Ashley Parker. She is a neuroscientist studying sensory motor processes and neuromechanisms in animals. Her work combines behavioral research with neurophysiological methods, such as EEG, to investigate brain function in large mammals like elephants. So, Ashley.
Am I supposed to see something here, or is it okay to not? I mean, it doesn't matter. Okay, I mean, I can just deal with it. Okay, I know it's been a long day, and I'm now the last talk, and I'm sure you're all exhausted. So I hope this will be at least easily digestible, and I can convince you that these squiggly lines are interesting. So yeah, I'm going to tell you about a very early ongoing work that we're doing. I'm from the Humboldt University in Berlin, and I'm working in the Berlin Zoo there. And we're trying to see whether we can get EEG, which you will all have heard of. It's that process where they put electrodes on your head and they try to read your brain waves. And we're trying to see if we can do this in elephants. So I don't think I need to convince... Oh, I should probably... Am I loud enough if I stand here? Oh, sorry. Okay, sorry, online people. I don't think I need to convince all of you that elephants are pretty remarkable animals. They are very family-orientated, especially the females lead these herds. They have very complex social dynamics, communication. But one of the things that also sets them apart from other animals is their trunk. And their trunk is used for all sorts of different activities. I often tell people that when I interact with elephants, it's almost like the rest of their body is not there. It's like you're just interacting with their trunk because it does everything. It smells you, it feels you, it's sensing the room, and they use it to drink, they use it to feed, they use it to socially communicate, problem solve, you name it. And I have a sweet video that was taken at our zoo, done by a colleague with Pa. Some of you might have seen it. She has learned to peel bananas. She does this on her own. So she doesn't like brown bananas. And so she removes the peel and just eats the pulp inside. And this is a very complex task that she's managed to achieve. And it's amazing dexterity with an organ or like a limb in a way that has so much power and yet so much dexterity. But in my lab, we're interested in the elephant brain, and specifically how the trunk and brain like work together. We have no idea where in the brain the trunk is, like the information of the trunk is being held, and this has to be a big part of the brain. It's so essential to this creature. But the one problem is that the brain is situated really deeply inside the elephant's head. Traditionally in neuroscience, if you want to study live brain recordings, you would either put a human in an fMRI machine, so a functional MRI machine. There's newer techniques. We can look at blood flow, but that also involves getting close to the brain. You can also stick electrodes in, or some people take the cap of the head off in rats and mice and see what's going on inside their brain. This is not possible with an elephant, obviously. So our best bet to get any sort of brain recordings from an elephant is with EEG. And so we teamed up with this gentleman, Dr. or Professor Klaus Graumann, who is an expert in mobile EEG. So essentially he does EEG in humans, but they're free to walk around. And this is essential because EEG essentially just measures voltage potential. And our brain emits these little voltage potentials all the time, but so does our muscle, and so do just moving your eyes left and right, and so does the heart, and so does the cables in the electricity lines, and also the cables of the EEG rig themselves. So you can imagine you get a lot of noise in the data you're trying to collect, and you've got to try tease out what is brain data versus other noisy data. And the problem is that humans typically will be told to sit very still, which is obviously a very difficult thing to do with an animal. And typically when they do EEG in animals, they are anesthetized, but that doesn't really work if you want to get EEG for a specific task where the animal needs to be awake. So we designed this elephant-specific EEG rig. So it's got 8 electrodes that we record from, some reference electrodes, and the back has an amplifier that means the elephant doesn't have to be tethered to a computer to collect the data, but instead we can Bluetooth this data to our computer. This is Anchali. She's the elephant we work with. She's 13 years old. We went for the option initially to see if we could get readings from the back, because this is where the skull is the thinnest, but it's also where a ligament sits to hold the head up. So it was a compromise. We tried the back at first. It's this 8 electrode array that we measure from. And then we get Anchali. So I guess the question is, like, how do you prove that what you get is brain recordings, right? And this is what we want. This is what we're setting out to do, to prove that we're getting it. And since we're interested in the trunk, you can do this by giving an animal or a human a stimulus that happens at random intervals. It's important that it's random. And in this case, we give her a vibration on that bar, and she puts her trunk on that bar. I'll show you a video in a minute. And this bar vibrates at random times, and it's a bit like if you were holding a cell phone, you would feel the vibrations in your hand. It's a similar level of vibration. And so she holds this, and as it vibrates, we're recording from her brain, and we know exactly when these vibrations happen, and then we would look for a predictable signal that comes after these vibrations in the brain. So I'll show you a quick video of how we do all of this. So this is Anchali. She's been trained over a long time to get this setup installed on her. She's very compliant with it. Often when I walk in, she starts kneeling down already to receive we put some paste on her head that helps stick on the electrodes. It makes it easier for us to take them off her without hurting her. And also the paste has got salts in it that makes the conduction of the voltage easier. And then she gets these very big electrodes stuck on her. It's completely painless, so there's no discomfort for her. And now we stick on the electrodes. We plug them into the electrode pads.
and we have to stick on the amplification box. And she's very patient through this whole activity. She, of course, gets rewarded constantly. And then we bring the box over. You'll see that she's keen to start, even though I'm not ready to start the vibrations. And we video it all the time, so we know what's kind of happening, when she's holding it, when she's not. And then she's got to hold this bar like this, and it's busy vibrating. You can't see that, obviously, but yeah, the vibration is similar to that of a cell phone. And then she gets rewarded in between. That's the bag of apples behind me. And yeah, so she'll get her snack reward. And from what I can tell, she never shows any discomfort or not willing to participate or cooperate with us. And if she does, which once happened, she wasn't in a good mood, we just aborted the experiment that day. So this is the result we get, which is essentially a lot of squiggly lines. You'll notice that there's 8 kind of rows. Those are each of the electrodes. And then there's time on the x-axis. You'll also notice these green lines. These are the points of vibration. So every time there's a vibration, it's measured. And as I said before, this is important because we then need to see if we can see a predictable pattern after each vibration. It's not going to exist after each vibration because there's going to be different levels of noise at different times, but the idea is you want to get as many of these vibration events as possible, and then you kind of cut the data out, making the time of the vibration time 0, and then you expect to see the same sort of pattern after the same number of milliseconds after this vibration event. And so what you get is something like this. So this would be each of the electrodes. And does this have a laser? No. And you would see some sort of pattern happening, and you would want to be seeing it at the same time. Roughly the same distance after this point here, which is when the vibration happens. And okay, this looks all right. We see, like in the top left one, that there's 2 peaks, or it goes down and up. We see it going up again on the next one down. We see something similar there. But what's important is that we want to see this then when we go the next day and do the same thing, right? We expect the brain to behave in the same way. And we were finding that this wasn't really the case. Like, we weren't seeing the same thing every time we went. I then switched it up and tried on the front of her head, and the signals were actually much cleaner, I think because there's less muscle on the front of her head. So we thought this might be promising, but again, wasn't really looking the same. And at this point, I'm kind of just don't really know what to do, because I was like, well, you know, maybe we're not ever gonna get it. Like, maybe it's just too far away from our signal for us to even get any signal, so I don't even know how deep to look. But then, very fortuitously, 2 weeks ago, we got an opportunity that Charlie was undergoing AI, artificial insemination, and they had to sedate her for this. And this was really beneficial because it meant we could put the electrodes on and she wasn't going to be moving. And also, when an animal or human is in a sedated state, we have expectations of certain brainwaves. And so we could then see what sort of results we got when she was sedated, and this would be the cleanest way we could see whether we were getting any brainwaves. So this was the data. We put electrodes on the back and the front because we wanted to optimize our opportunities. This was a super lucky situation that we could put the electrodes on her. And so the colors just indicate whether it was at the back or the front. And yeah, it doesn't look like anything special, but if you look closely, you see these repeated things. This is the heartbeat. This is not what we want, but we were getting the heartbeat. Interestingly, it's always at the back electrodes. This also makes sense. It's closer to the heart. But importantly, so now you can see we're collecting more than just potentially brainwaves. We're also collecting something like the heartbeat. And you want to remove these things. So you want to work out, like, okay, what is meaningful and what is not meaningful. And so you can do this analysis. So I'm just going to give you an analogy to understand it.
So if you're in a room of people, 5 minutes? Okay. So if you're in a room of 1 minute. 1 minute? Yeah. Okay. Okay, we'll skip the analogy. It's okay. But basically, with some fancy maths, we're able to break down these different electrodes, gather them all together, and pull out important components or important features. And so we can pull out, you can see here, the last feature is the heartbeat that's been pulled out. And then we can say, okay, well, what is happening in all these other components? So these are no longer channels, they're kind of like mixtures of channels, reshuffled and pulling out important aspects of the data. But then what we're interested in is what sort of waves are we seeing? What sort of patterns are we seeing? Are we seeing things that look like brain waves? More fancy maths, you can break complex waves like this, like an EEG, into simpler waves. You can see what frequencies it's made up of. Don't worry about it. It's just a very useful tool. And that means we can then plot on the bottom of this axis different frequencies, and we always express brainwaves in by their frequencies, and we can see how much power or how represented that frequency is in the data. The important thing to pick out here is these two peaks that are in the alpha wave, so the colors are represented back there, and the beta waves. And these are very good signatures of brain waves. So this, to us, gave us a really good indication that what we're getting is actually brain waves in Antrali, and this gives us a lot of hope to go back to our vibration studies and just dig a little deeper in order to see if we can find this evoked response potential. If we can see a signal to this vibration. I will skip my future directions, but essentially we want to try EEG in babies that have much smaller skulls. And my colleague who gave a poster, she works on sleep, and we think we have a possibility to look at sleep, and this would be amazing to tie together our two projects of whether REM sleep is happening or not, because they're very clear signature patterns in REM sleep in the brain. And with that, I would like to thank the zoo in Berlin and all the collaborators I've worked with as well.
Thank you, Ashley, for a wonderful presentation. Does anyone have a question for her? Yes.
Hi, yes, fascinating talk, thank you. Lots of questions, but I'm gonna keep it short to the, when you were recording live, so not under sedation, I was wondering, because just in the image, I thought, like you said, there might have been movement interference. I also wondered, the setup, there was quite a lot of metal around, so if you were getting any sort of metal interference as well, or did you record outside? And could you train her to stand completely still to remove the movement interference?
So metal, I don't think would cause an issue. It's more if there's electricity running through that metal. Yeah. Luckily, we know what frequency electricity is at, so we can remove that from the data automatically. And then in terms of her standing completely still, I think the vibration task is pretty good at keeping her still, because she's got a focus. She does still sway and stuff, but you can't control them clenching their jaw or slight head movements and things like that. I think it would be really hard. Yeah, I can see very clearly in the data when she moves, and then I'm just delete that. So there are periods where she's still. I think our issue is we need to kind of pool data sets together because we just don't get enough time with her to get, the sedation data was over an hour and a half, and normally the live data is 10 minutes, half of which she's moving too much, she's not actually touching the vibration stimulus. And so we would need to just pool sessions together, and we haven't done this yet, but this was what I think we kind of learned from the sedation data. Yeah.
Cool. I'll save my other questions for later. Thank you.
Sure.
Thank you. Any other questions for Ashley? Okay, thank you very much. Great presentation. Okay, so that brings us to the end parts for the in-person attendees. We will be starting the online poster session shortly. I just want to make a few announcements. So please remember to vote for best talk and best poster online and in person. I had said previously to submit the papers at the Savannah at 5 o'clock, but we have now the scavenger hunt, so you can do it by 5:30 or as soon as possible when you arrive for the gala night. Your silent auction bids as well, you can submit at 5:30 or as soon as you arrive at the gala night. And now, if you look in your bags, there is the map for the scavenger hunt. I had painstakingly circled where you're supposed to go. You can go at any order, and you can do it at teams of 2 or more people. Or you can go by yourself, but it's always nice to go in a team. So you have the map of where to go, and when you arrive at each station, there is a hint for where you find the questions, and then upon finding the questions, you can then circle what the answer is on your answer sheet. All right? So if you have any questions, I or Ruth or Philip can answer them for you. Okay, so Philip is going to moderate the online poster session. We want to thank you all so very much for your amazing attention and participation. And from our side, in person, this is the end of the 6th International Meeting on Zoo Research, Conservation, and Biodiversity. Thank you.